Right now, as you sit watching this, roughly 39 trillion bacteria are living inside your body. You are carrying more bacterial cells than human cells. Every single day, millions of people swallow a probiotic capsule, spending over $65 billion a year on a global industry built on the promise that adding good bacteria to your gut will improve your health. Most of those bacteria never survive. The majority of probiotic strains are dead before they ever reach your intestines. They are destroyed by stomach acid, dissolved by bile salts, and flushed out of your system within 48 hours. The few that do make it past those defenses enter a war zone. Your gut microbiome is a densely populated ecosystem where established bacterial colonies fight to the death to defend their territory. We need to look at what actually happens when billions of foreign microorganisms enter an ecosystem that has been evolving inside you since the moment you were born. This process engages your immune system, your metabolism, and a communication network so sophisticated that scientists now call your gut the second brain. Over the next few minutes, we are going to see exactly how probiotics function at the cellular level to determine what actually makes a supplement effective. Deep inside your large intestine lives a species of bacteria so ancient that it has been colonizing human guts for over 200,000 years. It does not exist in any probiotic supplement on the market, yet emerging research suggests it may be the factor that determines if a probiotic works. This ancient species is a critical link in your internal ecosystem. Welcome to BioState. To understand what probiotics walk into, we have to look at the scale of the gastrointestinal tract. It is a tube roughly 30 feet long. Stretched out flat, the surface area of your intestinal lining would cover an entire tennis court. Every square inch is coated in bacteria, containing up to 1,000 different species. Together, they weigh about 4.5 pounds, heavier than your brain. These bacteria are not just hitchhikers. They are active chemical factories. They produce essential vitamins that your body cannot make on its own. They manufacture neurotransmitters that regulate your mood and train your immune system to distinguish between harmless food and dangerous pathogens. When this ecosystem is balanced, digestion runs smoothly, your energy stays stable, and your immune system responds appropriately to threats without overreacting. When the gut microbiome falls out of balance, a condition called dysbiosis, the consequences ripple through your entire body. Research links dysbiosis to chronic bloating, irritable bowel syndrome, skin conditions like eczema, persistent fatigue, and brain fog. This chart, based on a study published in the journal Nature, shows that individuals with low microbial diversity have significantly higher rates of obesity, insulin resistance, and chronic low-grade inflammation. Modern life acts as a wrecking ball to this environment. Processed foods, artificial sweeteners, chronic stress, and poor sleep can devastate your bacterial populations in a matter of days. A single course of broad-spectrum antibiotics can wipe out 30% of your gut bacteria. Some studies suggest it takes up to 6 months for those populations to recover, and some species never return. When someone reaches for a probiotic, they are deploying microscopic soldiers into a severely degraded battlefield without understanding the terrain they are entering. A probiotic capsule typically contains somewhere between 1 billion and 100 billion colony-forming units, or CFU. That sounds like a massive number, but your existing microbiome contains roughly 38 trillion bacteria. As this visualization shows, even a high-dose probiotic represents less than 1,000th of 1% of your existing population. This creates a paradox. How could such a statistically small number of foreign bacteria make any difference in such a vast environment? The assumption that probiotics heal by permanently moving into your gut is mathematically and biologically impossible. Most probiotic bacteria do not permanently attach to your gut. They are travelers, not residents. Their value lies in the cascade of biological events they trigger as they pass through. They release signaling molecules that interact with your native bacteria and your own cellular structures. This temporary interaction is what actually alters your internal chemistry, rather than permanently colonizing the space. The success of a probiotic hinges on the biological chain reactions it can initiate within the brief 72-hour window after hitting your stomach. The moment you swallow a probiotic capsule, it enters one of the most hostile environments in the human body. Your stomach maintains a pH level between 1.5 and 3.5. That is acidic enough to dissolve metal. This is your body's first line of defense against biological invaders, and it is entirely indiscriminate. It destroys harmful pathogens and beneficial bacteria with the same lethal efficiency. Without protection, gelatin capsules dissolve in 30 minutes, eradicating up to 90% of probiotic bacteria. Notice the data change with advanced formulation. Enteric coatings shield the payload, allowing up to 80% to survive the acid and push forward. Consuming beneficial bacteria is a wasted effort if the physical delivery vehicle isn't engineered to survive this initial acid assault. Imagine a single lactobacillus bacterium that survived the acid, squeezing through a muscular valve called the pyloric sphincter and drop into the duodenum, the very first section of the small intestine. At roughly 2 micrometers long, this bacterium is about 50 times smaller than the width of a human hair. And waiting for it in the duodenum is a second hazard. The body floods this area with bile salts and pancreatic enzymes, chemical agents designed to break down dense fats and proteins from your diet. These same digestive chemicals immediately begin to erode and damage the delicate cell membranes of the surviving bacteria. It acts as a microscopic chemical rainstorm. Navigating the stomach acid is only the first hurdle. These organisms must possess the structural resilience to withstand severe chemical weathering before they can initiate any biological benefit. The bacteria resilient enough to survive that second wave drift deeper into the small intestine. Here, the contents of your gut are separated from your bloodstream by a delicate epithelial lining exactly 1 cell thick. Beneath the thin barrier is a dense network of specialized cells, the gut-associated lymphoid tissue, containing 70. By the 48-hour mark, the bacteria cross into the large intestine. The colon is the most densely populated microbial habitat on Earth, containing over 10 trillion bacteria per milliliter of content. Space and resources are scarce. The new probiotic arrivals immediately engage in competitive exclusion, fighting harmful resident bacteria for available nutrients and physical binding sites on the intestinal wall. They wage microscopic chemical warfare to secure that territory. Certain lactobacillus strains release lactic acid to lower the local pH. At the exact same time, others deploy targeted natural antibiotics called bacteriocins to aggressively destroy the cell membranes of competing pathogens. While securing territory, a secondary action begins. The probiotics start fermenting dietary fiber from your meals, breaking it down into metabolic fuel known as short-chain fatty acids. This diagram details the critical role of one specific fatty acid, butyrate. It is the primary food source for your colon cells. Without it, the cells shrink, allowing jagged toxins to slip through. When butyrate is applied, the cells expand and lock tightly together. When colon cells starve, the intestinal barrier weakens. Toxins and undigested food particles leak directly into the bloodstream, a condition widely known as leaky gut. These organisms act as microscopic chemical factories, manufacturing the exact fuel required to maintain the structural integrity of the intestinal seal. At 72 hours, the local repair work inside the gut continues, but the probiotics also initiate a systemic process that reaches far beyond the abdomen. Specific strains of gut bacteria actively manufacture critical neurotransmitters. They produce gamma-aminobutyric acid, along with dopamine and the precursors to serotonin. This stool data visualization compares serotonin distribution in the human body. Roughly 95% of your serotonin originates in the gut, with only about 5% produced in the brain. These chemical signals travel up a physical superhighway called the vagus nerve, a bundle of over 100,000 nerve fibers running from the abdomen into the brainstem. This bidirectional network means the temporary bacterial population in your gut is a constant, active participant in your neurological signaling. In just 3 days, a single properly formulated capsule navigates an acid bath, trains the immune system, fortifies the intestinal walls against toxins, and sends neurotransmitters racing up the spinal column to the brain. But these 72 hours of immune, metabolic, and neurological upgrades are merely the prologue. The real question is what happens to your biology when you repeat this cycle consistently for 30 days. For the first 72 hours, probiotic bacteria act as visitors. They interact with your immune system and compete with harmful microbes, but they do not permanently alter the landscape of your microbiome. That begins to shift around the 2-week mark. By day 30 of consistent supplementation, the changes become measurable across nearly every major system in your body. This data visualization shows the results of a 2022 meta-analysis published in the journal Nutrients. Across 14 trials, researchers found that daily probiotic use increased bacterial species richness by an average of 12 to 18%. This diversity creates stability. Much like a financial portfolio, a diversified microbiome is less vulnerable to a single strain crashing. While species richness increases, the actual composition of the microbiome also shifts. Populations of beneficial species like Bifidobacterium longum expand, while potentially harmful species like Clostridium perfringens decline. This rebalancing reduces the production of endotoxins. These are toxic compounds released by certain bacteria when they die, acting as the primary triggers for chronic, low-grade inflammation. This chart tracks the resulting drop in blood markers for inflammation. In multiple clinical trials, C-reactive protein levels decreased by 15 to 25%, with similar reductions seen in interleukin-6. By neutralizing endotoxin production, the gut biome removes a significant trigger for systemic inflammation. This shift reduces the internal stress associated with chronic conditions like heart disease and type 2 diabetes. These gut-level changes broadcast upward through the gut-brain axis, a physical communication pathway linking your digestive system to your central nervous system. An Oxford study found that after 4 weeks of taking Bifidobacterium longum, participants showed lower cortisol levels upon waking and performed better on cognitive tests measuring memory and sustained attention. This cross-section shows the primary mechanism, butyrate. Produced by probiotic bacteria, it strengthens the blood-brain barrier to protect the brain from circulating toxins. Simultaneously, serotonin precursors travel via the vagus nerve to the limbic system, helping restore chemical communication. The ripple reaches the skin as well. A study in the journal Beneficial Microbes found that 12 weeks of Lactobacillus rhamnosus supplementation reduced adult acne severity by over 40%. This occurs through the regulation of insulin-like growth factor 1. By lowering this hormone, the body dials down sebum production, addressing a biological root cause of acne. Ultimately, issues like adult acne or persistent anxiety are often the outward expressions of this internal disrupted chemical communication system. Your microbiome also influences your metabolism by dictating how you extract energy from food. Specific bacterial strains increase the production of the hormone GLP-1, which slows gastric emptying and improves how the body responds to insulin. This chart tracks a trial from the European Journal of Clinical Nutrition. Over 12 weeks, participants taking a multi-strain probiotic lost an average of 3.5 pounds, or 1.6 kilograms, more than the placebo group, without changing their diet or exercise. Your gut also regulates sleep by producing melatonin precursors and signaling the body's internal rhythms through fatty acids. This graph illustrates sleep data from a 2023 study. Participants taking Lactobacillus gasseri fell asleep an average of 12 minutes faster and experienced a 14% increase in deep sleep duration. Strengthening the gut ecosystem improves how the body extracts fuel during the waking hours and how it repairs itself at night. If clinical trials show such clear results for the skin, brain, and metabolism, why do so many individuals take probiotics and feel no difference at all? There is a significant gap between the measurable shifts documented in studies and the common, frustrating experience of feeling like the supplement isn't working. The reason for this discrepancy is a biological concept called colonization resistance. This diagram compares two gut environments. In environment B, an impenetrable wall of native bacteria prevents new probiotic strains from taking root. In environment A's open matrix, the incoming unit integrates, triggering the metabolic and cognitive improvements we've explored. In B, it simply fades away. A probiotic's impact is never determined by the capsule alone, but by the pre-existing biological landscape it is attempting to enter. Inside your digestive tract, your gut microbiome maintains a highly specialized internal security system. Researchers call this mechanism colonization resistance. Colonization resistance is the ability of your native bacteria to defend their territory. When a new species arrives, even a probiotic one, Your existing microbes work together to prevent the newcomer from establishing a foothold. Microbiome data reveals a consistent pattern. The more diverse and robust your gut ecosystem is, the more effectively it prevents probiotic supplements from having an impact. While many people view probiotics as an automatic biological upgrade, the gut's internal security system treats these supplement strains exactly like any other foreign arrival. In 2018, the Weizmann Institute of Science tested this by sampling the gut directly. After volunteers took a multi-strain probiotic, researchers used endoscopies and colonoscopies to see which bacteria actually stayed behind. In the first group, termed the persisters, the probiotic bacteria successfully colonized the digestive tract and triggered changes in local gene expression. In the second group, the resistors, the probiotic bacteria passed through the entire digestive tract without taking hold at all. Whether a participant was a persister or a resistor did not correlate with their diet, age, or genetics. The outcome was determined by pre-existing microbial diversity. In a healthy gut, a dense matrix of native bacteria physically blocks the newcomer, but in a depleted microbiome with specific gaps in its population, the supplement strains can easily attach and begin to function. Because probiotic success depends on an individual's unique biological baseline, universal health claims about these supplements are often inaccurate. Many people reflexively reach for probiotics immediately after finishing a course of antibiotics to help their gut recover. This chart tracks the recovery of the microbiome after antibiotics, and the results suggest a different approach is needed. In the Weizmann study, the group that took probiotics saw a 5-month delay in the return of their native bacteria. Contrast that with the group that took no supplements. Their native microbiome saw a rapid rebound, returning to its original baseline within 3 weeks. By flooding the empty terrain with foreign strains, the probiotics effectively occupied the space that the body's native ecosystem needed to repair itself. Probiotics are generally safe for healthy adults, but people with compromised immune systems, central venous catheters, or short bowel syndrome face documented risks of bloodstream infections like bacteremia. For most users, symptoms like gas or bloating in the first 2 weeks are simply signs that the microbiome is adjusting to the new arrivals. If that discomfort lasts longer than 2 weeks, the specific strains in the supplement are likely incompatible with your individual microbiome. To find the most important microbe in the human colon, we have to look at a species that has lived in human intestines for over 200,000 years, Faecalbacterium prausnitzii. It is a keystone species because it produces the majority of your gut's butyrate, the short-chain fatty acid that feeds your intestinal lining and regulates inflammation. You will not find this species in any supplement capsule because we currently lack the technology to mass produce it. Faecalbacterium prausnitzii dies within minutes of contact with air, making it nearly impossible to stabilize in a manufacturing facility using today's methods. This means the most vital organism for human gut health is entirely absent from the probiotic market. Probiotics do not function by permanently replacing the missing bacteria in your gut. Instead, they act as temporary residents. As they pass through, these supplement strains lower the internal pH and produce specific short-chain fatty acids. This temporary change in the environment creates the specific conditions your native Faecalbacterium prausnitzii needs to multiply and recover on its own. Probiotics are not replacement parts. They are environmental modifiers that prep the soil for your native species to return. When choosing a probiotic, prioritize multi-strain formulations that contain well-studied species of Lactobacillus and Bifidobacterium. Ensure the label lists exact strain designations and look for third-party testing to verify the bacteria are actually alive at the time of purchase. To minimize adjustment symptoms, start with a lower dose and increase it gradually over several days. However, the primary driver of gut health is feeding the bacteria you already have. A diet rich in diverse fibers, fermented foods, and polyphenols provides the fuel your native microbes need. Your microbiome is a living ecosystem that requires the right environmental conditions to flourish, rather than a machine requiring replacement components. This is general education, not medical advice. Consult a clinician before making health changes. If this changed how you think about your body, subscribe to BioState for more.